you that may have been wondering how I got on at the Oxford 1910 annual members show. As you can see, these are the two birds that did well for me at the show. I won both novice any age and novice young bird, so quite pleased with that outcome. Hello and welcome to part 2 of my introduction to Budgerigar genetics. In part 1 we learned some of the key terms that we're going to need to understand how the colour mutation moves from one generation of Budgerigar into the next. We learnt about cells and about chromosomes and genes that sit within them. We also learnt some of those key words such as the locus, the allelese, heterozygous and homozygous. If you missed part 1 or feel you need refreshing on some of those terms, please click on the link and I'll see you when you get back. Well, in this second video, in the introduction to Budgerigar genetics, we're going to look at what happens when the new bird inherits the genes from the two parent birds and how these interact, and how this will then enable us to help predict the expected colour outcomes from any particular two pairings. Before we do that though, I'd like to introduce you to this person here. His name is Gregor Mendel. Gregor Mendel, who is known as the father of modern genetics, began investigating the hereditary traits in plants. He mostly studied pea plants. He looked at the traits that appeared to be independent of other hereditary, including the colour of the flowers. Between 1856 and 1863, Mendel cultivated and tested some 28,000 plants. His study showed that 1 in 4 plants had purebred recessive traits, 2 out of 4 had hybrid, and 1 out of 4 had prebred dominant. His experiment led him to make some generalisations known as the law of segregation and the law of independent assortment, which later become known as Mendel's laws of inheritance. So why is Gregor Mendel important to us? Well, for the purposes of this introduction to Budgerigar genetics, all of the colour predictions that we'll be making will be based on Mendel's laws of inheritance. There is one variety of Budgerigar where there's currently some discussion about whether it follows those laws, and maybe we'll touch on that in a future video. But for now, the laws hold true. And when you hear others around you talking about Mendelian laws of inheritance, then it's Gregor Mendel is who they are talking about. What I want to do now is just move quickly over to the computer and we can discuss what it was that Gregor Mendel found with his peas.
Well, here we are at the computer, and from my very brief introduction of the work of Gregor Mendel, you'll already notice and have heard some of the terms that we continue to use today when we're talking about Budgerigar genetics, such as dominant and recessive. So what I'm going to do now is introduce you to a simple technique that allows you to start making those predictions on pairing once you know some of the genetics or the genetic makeup of either the plant in this case or the Budgerigar, as we will see in a little while further. So one of the key tools that we're going to use is this thing here that's called a Punnett Square, which helps us to look at those possible predictions and the percentage outcome that we're likely to have. It's fairly simple to understand. Along the top here we have what I referred to as plant 1, but it's just one of the parent either birds or plants in this case, and we have plant 2, which is the second parent plant or bird. Underneath each of these two we have 2 smaller boxes that in this case both contain W's. These boxes represent the possible alleleses of each of the chromosomes. So and as we know that only one of the chromosomes, or one set of chromosomes, is passed on to the offspring from each of the parents, giving us 2. So in this case we've got a red plant that contains 2 chromosomes, which I've called 2 alleles, of which we've called W, and down here we've got 2 alleles which are marked by the lowercase double. This brings me to one of the common features, or one of the common rules that we use when we are naming genes. They're normally named after the mutation. So in this case the parent plant, which was red, the gene mutated to produce a different allelase that produced a white flower. So in this case the gene would be called the white gene, and as is customary it's been reduced to just 1 or 2 letters. Sometimes we have a number at the end of it, but 1 or 2 letters, and I've called it W, so W stands for the white gene. You'll also notice that I've placed them differently in terms of the upper and lower case. So what this stands for is it's common practice if a gene or if a particular allelase is dominant, then to name that with an uppercase letter, and if it's recessive to name it with a lowercase letter. So in this case the red allelase is dominant over the white allelase. This becomes more apparent as I go through and we start to use the 4 other squares here in the centre. So simple way to use it is that we're going to bring 1 down from each side to fill into this square. So in this case there's a uppercase W, and across here there's a lowercase w. And over here again we've got an uppercase W and a lowercase w. And this pattern is going to be repeated, as we can see, because both of the parents are homozygous, so we end up with the same in each of the 4 squares. So in each of the 4 squares we're going to have possible outcomes of the pairing of the 2 parent plants. And here we can see here that we now have a heterozygous offspring, in that it contains 2 different alleleases. One is the dominant allelase and one of the recessive allelase, and we know that the dominant allelase is red, and therefore each of these possible pairings, or each of the 2 possible pairings, will produce 100% white plant, 100% red plant, because red being dominant. So each of the pairing between this particular plant 1 and plant 2 will produce offspring that are 100% red. But now let's see what would happen if we paired 2 of the offspring together, so 2 of the offspring plants together. I'll quickly flick over to the next spreadsheet, which contains a separate Punnett, which is already a Punnett Square, which is already set up to look at the pairing between 2 of those. So again we've got a red plant because it contains one of the dominant red alleleases and also the mutated white allelase that is recessive, and plant 2 containing the same, a recessive white allelase and the dominant red allelase. So what happens now when we pair them? We'll end up with a slightly different result in each of the 4 squares. So in this square here, once again we're going to bring the uppercase W down, and then coming across we're going to bring the lowercase w across. This one here we've got a lowercase w coming from the top plant and a lowercase w coming from the the left hand side plant. Down here we've got a uppercase W coming from the top plant and a uppercase W coming from the second plant, and in here we've got a lowercase w coming from the top plant and an uppercase W coming from plant 2. So as we can see, we've got different outcomes this time from pairing these 2 plants together. We have 2 plants that are heterozygous, and that they carry both the non-mutated allelase and the mutated allelase, and then we have 2 plants or 2 possible outcomes that are homozygous, and they contain either the complete mutated allelase or the 2 non-mutated alleleases. So what would these look like? Well, we already know that these 2 are going to produce red plants because they contain the dominant non-mutated allelase, and this one here will also be red because it contains both the dominant red alleleases. However, this one over here would be white because it contains both of the two mutated and recessive white alleles. So in this particular pairing we would end up with 75% of the offspring being red and 25 of the offspring, or 25% of the offspring being white. So there's an outcome that we would know. So if we planted the 4 seeds, based on just on percentages, we would expect 3 red plants and 1 white plant. Of course, that's just a percentage outcome, and it won't always happen in that way. But if you over a period of time, that's what we would expect the outcome to be. This also produces something fairly interesting in that we have 2 birds here with different genetic two plants here, would be the same for a bird, but 2 plants with different genetic makeups that actually look identical. So they would both look, so both plants would look red, and just by looking at them you wouldn't know whether one of them was carrying the recessive white gene. So this actually introduces a new term into the study of the traits and how they're passed on, and that is what a plant or bird looks like. So it's phenotype as opposed to the genes that it takes, which is known as its genotype. So in the case of our 4 possible outcomes here, we have 2 homozygous birds that which with a phenotype that reflects exactly their genes, whereas we have 2 heterozygous birds here whose phenotype is different from their genotype. It's common for us to refer to this when we have a plant, or particularly a budgerigar. You'll hear often people refer to it as being split for something. So in this case, this plant here in this top corner here would be red split for white, and we often hear people talking about something being green and split for blue, or being green and split for So that's what we mean by split. It merely means that its genotype is different from the phenotype. So just to recap on that, then the phenotype is what the bird looks like outwardly, and the genotype is how the genes that it actually contains. And 2 birds can have the same phenotype but have different genotypes. Well, now we've looked at our hypothetical crossing of 2 peas or 2 pea plants, let's have a think about how this reflects to the pair of birds that we started off with in the first video, so in the first introductory video, where we crossed a sky blue cockbird with a light green hen.
And here we are back at the cages, and we'll look at how the sky blue and the light green began to produce, or can produce, a white lacewing that we've got here, or at least we'll start to understand that when we look at the next set of Punnett squares. But in the meantime, before I do that, what I'd like to do is very briefly discuss the differences between a light blue and a sky blue and a light green bird, the green and the blue series birds. This may sound obvious, one's green and one's blue, but there are some other differences which help us understand some of the other varieties as we move through this series. So as we said, if we look at the 2 pictures here, you note the obvious difference is that the blue bird is mainly blue, and the green bird green series is mainly green. However, there are some subtle differences between the 2. The first of these is that if you look at the face and the wing area, the area on the light green that is yellow is white on the sky blue. So in effect what's happening here, what the mutation is actually doing, is changing the pigment that produces the yellow in the bird. So all those parts that were yellow are now white in the blue series, and in the green series clearly they're yellow because the mutation hasn't happened. And if we all know that if you mix blue and yellow together you make green, so we can now understand why the blue series bird has a blue body. It's in fact the green body minus the yellow pigment. So because green minus yellow equals blue. So that's important to understand, that in fact this bird here, although it's white, is clearly a blue series bird. So it must have the mutated blue gene in it. Let's go back over to the computer now and look at how this might have occurred.
So here we are back at the computer with our new Punnett Square that shows the crossing between our sky blue cock and our light green hen. We'll start off with the sky blue cock. Now we know that the blue allelese is recessive, so therefore in order for this bird to be phenotype sky blue, it must contain both of the blue series mutations. So I've put the lowercase blue recessive mutation in both of the two allelese squares. There's a 1 next to it because this particular gene is mutated more than once, but we'll come on to that in a later video. As for the green series bird, well, we know this could either be homozygous or heterozygous. For the time being, we've put it down as being homozygous, with both the alleleses being the dominant unmutated green type allelese. So let's have a quick look at what the outcome would be if we paired this and see if there's any possibility of producing the blue series bird that we want to produce. So we'll just do exactly the same as we did before, bring the b, the small lowercase b and the 1 down from the top, and then bring across the uppercase b and the 1 from the side. And the same again here. And now same down here. So there we go. All 4 outcomes are exactly the same as we saw in the previous one with the red and white P crossing. So all the outcomes will be heterozygous with 1 of the mutated recessive blue genes and 1 allelese and 1 with the dominant green allelese, which means that it will be 100% green birds. All the green birds, however, will be phenotype green, but genotype will be green split for blue. So under this pairing, we now know that this wasn't the gene pairing that we had, because we are certain that we produced a blue series bird. So let's have a look at how we could produce the blue series bird. We'll move over to a new spreadsheet here, and this time we've got the green hen is going to be heterozygous with 1 dominant allelese and 1 recessive allelese. So let's have a look at what the outcome would be if we paired these. So again, doing exactly the same thing, we've got the small b and the uppercase B in that square, and we've got exactly the same here, so a small b with the uppercase B1 in that square there. Down here, however, we've got a lowercase b and a lowercase b. Didn't quite work, it predicted itself. And across here we'll have a lowercase b bringing down from the top, and another lowercase b being brought down from the side. So in this case we've got 2 different outcomes. So on the top here we've got the same as we had before, with a bird that's going to contain 1 of the allelese for the dominant green and 1 for the recessive blue. So we will have a green bird across the top here. Both the 2 outcomes here would be green. Down here, however, we've got 2 recessive allelese, which means it will produce the blue series bird. So in this particular pairing we would have 50% blue birds and 50% green birds. So we now know that the pairing that produced the lacewing the green hen must have been green split for blue. So it must have phenotype must have been green with the genotype green split for blue. It still doesn't account for why we produced a lacewing, but it does account for how we got a blue series bird from the particular pairing. So we know something now, that if we had 2 blue series birds, we would never ever produce any green series birds from it. So whereas if we have 2 green series birds, we could actually produce a blue series bird from that. So that's about all we've got time for in this particular video.
Well, thank you for watching part 2 of my introduction to Budgerigar genetics, and I look forward to seeing you all again in part 3. In part 3 we'll begin to explore the semi-dominant genes such as the spangle and the dark colour genes. So hopefully see you then. Please do like, comment and subscribe.